visualizer in Chromovirus. This work was mainly performed during the pandemic shutdown in 2020, and the research is still ongoing. In March 2020, my school in Boston, USA, was canceled, and I was restricted at home. I wondered what was special about coronaviruses and how we could eradicate them. So I Googled online and found many coronavirus DNA sequences of the NCBF virus habitat. I was taking computer science at that time, and I thought it would be nice to plot the DNA sequences and visualize the key features. So I developed a DNA nucleotide footprint plotter and was excited to see the geometric differences between coronavirus and other viruses. I further developed a peptide visualizer, which displayed the characteristics of peptides. In the next half hour, I'm going to talk about the two tools in detail. Okay, so my talk consists of three things. First, I'm going to talk about coronavirus in general. Then I'm going to introduce my first tool, the DNA nucleotide footprint plotter. And finally, I'm going to introduce my second tool, the peptide visualizer. Let's start with an introduction of coronavirus in general. So as you can see here, this slide is a transmission electron microscope image of a coronavirus. We can see the Krem shape. And then on this image, here is an animated photo of coronavirus proteins. The spikes are mainly made of S protein, they're also membrane protein, envelope protein, nucleocapsid protein. And yeah, so all these proteins are abbreviated on the screen. So I'm going to be referencing myself. S, M, E, and N. So S, M, E, and N proteins are all called structural proteins, and the genome of coronaviruses are about 26 to 32,000 base pairs long. The coding region of the structural proteins are after 20,000 base pairs. So the first 20,000 base pairs of the genome code non-structural proteins, including NSP1 to NSP16. They include important proteins directing coronavirus RNA synthesis process. Some functions of proteins are viral replication, inhibition of translation machinery, and production of immune defense factors. And those are the functions of structural proteins. The third group of coronavirus proteins are accessory proteins. They are related to immune evasion mechanisms and determine the distinctive host strain, virulence, and mortality rate of each coronavirus. The genes coding accessory proteins are located at 3' of the viral gene. So now that I've talked about the basics of coronavirus, let's move on to the second section of my talk, where I'm going to talk about my tool, which is the DNA nucleotide footprint plotter. So coronaviruses are RNA viruses, but people reverse transcribe RNA to cDNA in order to sequence them. We can apply the DNA footprint plotter to study the key features of coronaviruses. They're saying that a picture is worth 1,000 words. So I decided to convert the coronavirus genome sequences to shapes. What I mean by this is that the DNA footprint plots. I use Java to plot the 4 nucleotides in 4 directions, right, left, up, and down. There are 3 different ways to do this: a tag, a DAC, and a cat. In the A tag plot, I plotted A to the right, T up, G left, and C down. In the A DAC, I plotted A right, G up, C left, and T down. And then in the A cap, I plotted A right, C up, T left, and G down. We can see that the shapes of the DNA footprint are different using the group plotting options. In the current research, I use A cap, and you can see in the image the shape of a coronavirus footprint looks like a fishing pole. Here is the Java flowchart of A caps. So to begin, The tool reads in coronavirus sequencing data. If the nucleotide is A, I let X plus 1, so the line segment moves to the right. If the nucleotide is T, I let X minus 1, so the line moves to the left. If the nucleotide is C, I let Y plus 1, so the line segment moves up. If the nucleotide is G, I let Y minus 1. So the line segment moves down if Y is minus 1. After all the nucleotides are plotted, the program ends. The DNA footprint plotter can distinguish different types of viruses. The first two plots are coronavirus plots. They look like fish tanks. Third plot is HIV, and the fourth plot is influenza. From DNA shapes, we can tell the types of viruses, and it has potential values to help diagnosis. The DNA footprint plotter not only can distinguish different types of viruses, but it can distinguish coronavirus subspecies. There are different types of coronaviruses, with OC43, HPV1, MERS, which is known as the Middle East strain, and SARS. OC43 and HPV1 cause mild symptoms, while MERS and SARS can cause more severe symptoms. We can see that the footprints are different among these different types of viruses. The shapes are similar within the same coronavirus types, but there are subtle differences. This has the value of facilitating vaccine development. By targeting the similar regions across different mutation strains, one vaccine might be able to fight different COVID variants. The DNA footprint plotter is very good at detecting large-scale structural variations such as recombination. I cut a part of the influenza virus sequence and a part of the coronavirus sequence from sequencing FASTA files, and I put them together into one FASTA file to simulate recombination. We can see DNA footprint plotter can clearly display this recombination in the chimera genome. See, this part is the influenza virus, and this part is coronavirus sequence and the recombined plots. Even if the DNA footprint plotter is built for structural variation detection, it can also display single nucleotide mutations. I developed an enhanced version that made the plotting interactive. If you drag your mouse over a specific position, the tool can tell the location, the genotype, and the corresponding amino acid. It also colors the 4 structural proteins in different colors, so that users can easily tell the locations of these proteins. I'm still working on this script to further enhance the user interface and the display, but it's very good as it is. I further enhanced my tool to compare coronavirus subspecies also. So, for example, I can compare coronavirus mutants to different colors, and I can overlay them on top of each other like the plot on the left. I also subtract the same regions between regions and only display the regions that are different like the plot on the right. Now, let's summarize what I've been studying. So, the DNA footprint plotter can display key features of the coronavirus genome. It can also display the similarities and differences in mutant strains. The plotter is good for visualizing structural characteristics. It can show simple nucleotide changes and information. And then if we look at applications, it can go beyond coronavirus, and we can apply this to other viruses, genes, and sections of eukaryote genomes. If we combine the plotter with other tools such as FLAST and phylogenetic tools, the DNA footprint plotter can directly capture the key features of DNA sequence. So on to the third part of my presentation. So after developing the plotter, the DNA footprint plotter, I thought it would be nice to visualize the key features of proteins also. So I developed the peptide visualizer. I'm going to talk about that in the next slides. Amino acids are the building blocks of proteins, right? And they contribute to different protein functions depending on whether they are positive or negative charges, or whether they are polar or non-polar, if the amino acid does not carry charges overall. By displaying different amino acids in different colors, and displaying charges, polarity, and molecular weight using colors and geometric shapes, the peptide visualizer demonstrates protein characteristics in a novel and straightforward way. The basic idea is to color each amino acid using different colors following the RGB convention. This is explained in the last column of the table. We can turn the line segments to different angles depending on the charges and polarities. I'll explain more details in the next slide. So here's the peptide visualized plotting criteria. So the length of the segment is proportional to the molecular weight of the amino acid. If it's heavier, then the segment's going to be longer. And then the color follows the amino acid color RGB convention. For positively charged amino acids, the segment angle turns clockwise by theta 1 degrees. For negatively charged amino acids, the segment angle turns counterclockwise by theta 1 degrees. Polar amino acids turn theta 2 degrees according to the original orientation, and non-polar amino acids turn by a smaller angle, which is theta 0. The users have options to adjust the above parameters according to the need of specific effects of different files. Currently, we define theta 0, theta 1, theta 2, and k to those constants on the screen. And so, and just for clarification, k is the parameter to determine if we're rotating clockwise or counterclockwise. And k represents the charge, as I just said. Here's the flowchart of peptide visualizer. I coded the visualizer also using Java. After begin, we put peptide sequences and we define parameters such as evaluate thetas. Then we move through each amino acid in the sequence and draw line segments. Theta is the cumulative sum of theta 0, theta 1, and theta 2. The sign is determined by k with values 1 or -1, which represents either positive or negative term. We then determine the values of x coordinates and y coordinates by taking the intersection of cosine and sine. After each amino acid is plotted, the program finishes. The peptide visualizer displays different shapes and different shapes. An example is displayed in the current slide. We can see the nucleocapsid protein and matrix protein of the bat chromovirus, and we can see that they have different shapes. Peptide plotters can distinguish proteins from different coronavirus species. The RBD region of S proteins are a region that many vaccines are designed to target. This slide shows amino acids 300 to 600 covering the RBD region of F proteins of different coronavirus types, including OC43, HPUI, MERS, and SARS. We can see the peptide characteristics are different across these different types of coronavirus. Within the same coronavirus type, there are similarities and subtle differences between different types. So we've already established that the DNA footprint plotters could explain structural variations. And we've also established that the peptide visualizer can enhance the presentation of functional changes at the amino acid level. We can see the DNA footprint is quite similar comparing the two leading coronaviruses. Of course, we can show differences, but at the peptide level, the potential functional changes at the single amino acid level is emphasized by the peptide visualizer. It's more sensitive to others. The peptide visualizer has broad application potential. It can also plot beyond coronavirus proteins. The current slide contains sample plots of plasmid and yeast proteins. The tool can be applied to plot eukaryotic proteins such as human proteins. There are many tools to display protein sequence changes such as blast and protein structures such as time. But the peptide visualizer emphasizes the features of the peptide inside of sequences or structures. For example, BLAST can tell you the changes between peptide sequences, but the potential functional changes are not as intuitive. For example, both lysine and arginine are positively charged. Aspartate is negatively charged. If using BLAST, lysine to arginine change, the lysine to arginine change is displayed similarly as the lysine to aspartate change. This is the change of a letter from K to R and K to D. But if we use the peptide plotter, the K to R mutation is displayed very differently from the K to D mutation due to the changes of charges, molecular weight, and color. Compared with Pymol, the potential functional impact is enhanced by the peptide visualization instead of the structure. So my research was published in 2 journals. One was Informatics and Medicine Unlocked, and the other one was Diagnostic Pathology. You can see in the red box. If you want the source code, check the green box, or my GitHub link is in there. So thank you everyone for listening to my talk. I hope that I can be in person to answer your questions, but I'm still in school and can't attend the conference in person. Feel free to email me at scram@bbns.org. It's the blue underline link on the slideshow. If you guys have any questions or suggestions, and I will make sure to address them. Happy holidays, everyone.